to introduce the speaker for today's special COVID-19 session, Dr. Josep Maria Llibre. Please mute the microphones. Thank you. Dr. Llibre is a renowned international scientist in the virology area, and since the beginning of the COVID-19 epidemic in Spain, this year in February, he has been involved in the front line of medical assistance of hospitalized patients with COVID and in clinical research as well. Dr. Llibre received his medical degree at the Universitat de Barcelona and obtained the specialty in internal medicine along with a PhD at the Universitat Autònoma de Barcelona. In 1987, at the beginning of the HIV epidemic in Spain, Dr. Llibre started caring for patients affected with HIV and AIDS. His main areas of interest in HIV medicine include the clinical implications of HIV-1 resistance, hepatitis C co-infection, with a special interest in the safety and efficacy of the antiretroviral drugs, and pharmacoeconomics as well. Dr. Llibre has been a member of the expert panel in the Spanish Society of Infectious Diseases and Clinical Microbiology, defining the HIV antiretroviral treatment guidelines, and also expert advisor in the Catalan Health Institute on antiretroviral treatment and hepatitis C as well. He has published widely in journals, book chapters, medical websites, and collaborates as an editor or peer reviewing with many top international medical journals. He's involved also in HIV medical educational training in Spain and other European countries. So please join me in welcoming Josep Maria Llibre, currently at Hospital Trias i Pujol, and collaborator in the group led by Dr. Clotet, is the team of reference for COVID-19 research in Catalonia. At the end of the presentation, we will have 10 to 15 minutes for questions. You can write your questions in the chat box or leave a note saying that you wish to speak. We will take questions in order of arrival. Please, Josep Maria.
Thank you very much, Dolores, for this nice introduction. This is Josep Llibre. I work in Barcelona in the University Hospital Germans Trias in the Infectious Diseases Department. I'm usually an HIV physician, but since this bad 2020 year, I have been forced to go to the COVID field. Thank you for this invitation to Clarivate. Actually, this is a poisoned invitation because it's not an easy field, as you're going to see. Just to summarize, I'm going to tell you that there's no single drug in the COVID therapy with all the data concordant, robust, and clear, suggesting or pointing to an incredible efficacy, especially in antiviral activity. There are significant weekly changes in knowledge. This doesn't happen in many fields in medicine, so everything changes, I would say every day, but every week in this field. And finally, the evaluation of what I'm going to say and the understanding is on my own. So this is going to be challenging. We'll try to give you a flavor of the HIV treatment and how the things have gone in this field. This is our unexpected invitee, the SARS-CoV-2. This is a beta coronavirus. This is a colored electron microscopy photograph with the virus showing the spikes in the membrane. You know very well the spikes because most of the vaccines and treatments are directed against the spike. And then you have the nucleocapsid like a nest of eggs inside the virus. Okay, this is how the world is going just today, right now. This is 4 weeks ago. We have more than 42 million cases along the world and more than 1 million. We are approaching 1,200,000 deaths. And approximately the overall mortality is 2 to 3%. But you know that cases that go into the hospital, the mortality reaches 5 to 10%. And the pace that the disease has nowadays in the world is 100,000 cases per day and 5,000 deaths per day. This is what we are declaring since many, many weeks ago. SARS-CoV-2 is actually a SARS-CoV-1 that has experienced an increase in pathogenicity. You know that the coronaviruses can mutate, but they usually don't evolve in nature only with mutations, but with recombination. They have recombination of big chunks of the virus, and they can increase the pathogenicity and the transmissibility. You know that there have been 3 epidemics of coronaviruses in the world. The first one was SARS-CoV-1 in 2003. The second one was MERS in the Middle East, 2010. And this one in 2020 is SARS-CoV-2, which is very similar to SARS-CoV-1, which was the first one. And the virus has virtually experienced an increase in pathogenicity. And this is important because these viruses can mutate, and this can have an impact against both antibodies and against drugs. Okay, the rhythm of the knowledge, it's outrageous. It's very difficult to follow the science in this field. This is something new. We were not aware before the coronavirus epidemic. It's the preprint publications. We are very used to go to journals, to medical journals, but it was quite unusual to have preprints. With this disease, this is the number of preprints available in medRxiv or in bioRxiv, approaching 10,000, so it's incredible. And if you go to PubMed and you write COVID-19 or SARS-CoV-2, you will receive more than 66,000 results. Sorry, we got noise. Someone should mute. Okay. No, that we have a noise. Someone should mute because otherwise. Okay. Thank you. These are the timelines. If you do a comparison between the HIV research and the SARS-CoV-2 research, just to let you know that it took 3 years in HIV. That was a very quick science field. It took 3 years from the identification of HIV to have the first drug, which was ACT. It took 5 months with SARS to have the first drug, which approved, that has been remdesivir, as you know. So the pace of development in this disease is amazing. Remember that we only knew this virus for 10 months, so we are very new to this infection. Let's go to the clinical field and the implications for treatment. This is a simplification of the clinical phases of COVID-19 disease. We established 3 stages. The first one is the infection. You have something very similar to a flu or to an influenza: fever, headache, cough, diarrhea, anosmia, dysgeusia. This happens for several days, and some people, most people will recover, but some people will go to the involvement of some organ, usually the lung, and they have a pneumonia. This is the pulmonary phase. It's still infectious or mainly infectious. It's the second stage, the pulmonary phase, that can involve any organ, virtually any organ in the body. And then most people will recover, but some people go to a hyperinflammation phase. This is the latest phase, and it's the phase where we see the most severe changes and the mortality. It's like a cytokine release syndrome. It's the cytokine storm. As you can understand, in real life, the phases are not so split. Everything is mixed, so there's a lot of overlap. But this should be a nice evolution of a typical case. But obviously, we see some diseases that are severe from the beginning, or that have a lot of hyperinflammation from the beginning. So this can be very overlapped. And we have 2 kinds of treatment. The first one is antivirals. This is a virus, so we have antivirals that can be drugs or antibodies. And we have immunomodulators. These are treatments that try to target or to slow down the hyperinflammation state. As you know, most of the people have an asymptomatic disease. They recover spontaneously. They have oligosymptomatic or a very low degree disease, similar to a flu. This happens in than 50% of the patients, so it can be unnoticed or with very few symptoms. But in some people, as we said before, you can have a lung involvement, you can have a pneumonia, bilateral pneumonia, and this can be devastating. This can be a very severe disease. People who go to the hospital and get admitted have a mortality close to 5 to 10%. This is a typical case of a previously healthy 20-year woman with a complete lung dysfunction that went to a bilateral lung transplantation. The thoracic surgeon said that that was the lung most destroyed that they had ever seen in their career. So this can be a devastating disease, and that's very important to remind always. And it's one of the reasons to understand why we went into a treatment with so low degree of scientific evidence. This is a typical picture of the cytokine storm. It's a massive spread of cytokines into the blood vessels that cause inflammation in the lung, but everywhere. They can cause a coagulopathy and lung involvement in the kidneys, in the heart, in the brain, in any organ in the body. Usually, when we have an infection, this should be a respiratory tract. This is the airway. These are the epithelial cells, and this should be a blood vessel. And usually, when something external that can be dangerous, like a virus, goes to the epithelial cells, you know that it attaches to the angiotensin converting enzyme 2 is a surface receptor that we have in most cells in the body, all the primates. It attaches to this receptor, it goes inside the cell, and then this releases cytokines that will get all our troops that have a response against this infection. That includes cytokines, cells, including the dendritic cells, T cells, macrophages, monocytes, and this causes a reaction against the infection and clears the infection, and you get cured. What happens in this disease and many other diseases is that sometimes this immune response is out of control. It's exaggerated, and it causes disease itself. So instead of removing the virus or curing the disease, it can cause itself an intrinsic disease. And so we thought that it could be a nice idea to block this exaggerated immune response. Obviously, in the immune response, we have many different pathways. Every pathway with every cell, neutrophils, macrophages, antigen-presenting cells that are the dendritic cells, CD4, CD8s, all the regulatory cells, they have a pathway that respond to some cytokines, and we have drugs that block all these cytokines. That's why we try to develop drugs that can block all these pathways. And it looks like right in the middle of most of these pathways of inflammation, it's interleukin-6. That's important because we have antagonists of interleukin-6. The first one we tried, as you all know, is tocilizumab, but we also have cetuximab, sarilumab, and many other monoclonal antibodies that can block interleukin-6. We can measure interleukin-6 easily in the blood. So in patients with an inflammatory picture with very high levels of interleukin-6, we thought it could be a nice idea to block this pathway of this exaggerated immune response. This is not so clear, and there are some analyses showing that if you compare severe or critical COVID-19 against other inflammatory syndromes like the cytokine release syndrome associated with CAR-T in the treatment of neoplasia of cancer, or with bacterial sepsis, or with AIDS-related acute respiratory distress syndrome, not related to COVID, so usually related to bacteria, the levels of inflammation are not the highest ones in COVID. So maybe we were wrong thinking that this exaggerated immune response was so important, because when you monitor the interleukin-6 levels, you can see that the higher ones are in the cytokine release syndrome or in the acute respiratory distress syndrome not related to COVID, and the levels are lower in COVID, so it's not so important. And the only thing which is really increased in COVID, it's D-dimer. D-dimer, as you all know, it's related with the coagulopathy, with thromboembolia, which is very common, as you know, in COVID. Lung embolism, deep vein thrombosis, this is very common. So many authors have now claimed that maybe that there's no increased immune modulator, cytokine storm release syndrome, and this could be just a part of the disease, and maybe we were wrong in directing treatments to this part of the disease. This is a picture. This is COVID. This is the acute respiratory distress syndrome. This is the T cell-induced cytokine release syndrome, and this is sepsis. And in all the models, you have the cellular compartment, the interstitial compartment, and the blood compartment. And you can see that the only thing that is really increased in COVID is D-dimer. There's not such a difference in interleukin-6, in tumor necrosis factor, or in other chemokines. So it's sure that the coagulopathy is a central part of COVID, but the other thing of the cytokine storm is much debatable. Okay, let's go to the treatment. And the first thing we have to understand is why things went this way. And the reason is quite simple. The treatment was panic-stricken therapy. We were facing a new disease. We initially believed that everyone was potentially susceptible, every one of us was potentially susceptible to infection. The disease is easily spreadable because this is a respiratory virus, so it can be transmitted easily with aerosols and droplets. Mortality is high if you have a moderate to severe disease, up to 10%. There was no proven treatment initially, and there was no vaccine. So the reason to start treatments with such a low level of evidence was panic. This is the usual way we develop drugs, and I think you know this better than me. In medicine, we start with molecular docking studies in the lab, in vitro studies, animal trials, preclinical human studies. Then in humans, we go to phase 1, phase 2, A and B, phase 3, and usually physicians are only familiar to drugs with phase 3 data. This takes usually 8 to 10 years. We didn't have 8 to 10 years to develop new drugs against SARS-CoV-2. So we went to use repurposed drugs. This means drugs that were in the market, that were available. You can save all the toxicity studies, so all the in vitro, all the animal, all the phase 1 and phase 2, because you know the dose. And you can go straight to use these drugs in the treatment with a compassionate use. So patients signing an informed consent. And despite this, there came up many small pilot observational studies without control arms. And this is how everything started. We were faced with pressing the do nothing button, or the button of do something that could be useless or could even be harmful. And we didn't know at the beginning. And that was not an easy choice, as you can understand easily. So why were we forced to use drugs with only docking information? Usually clinicians are not familiar with docking information. This, you know that drugs in docking studies, they have a library of 3D information. And when they know the receptor, they go to the library and they choose drugs that theoretically could attach properly to the receptor, to the pocket, to the infrastructure of this receptor. We now knew that the efficacy of these predictions is approximately 50%. So it's like flipping a coin. So it was not so a wise decision to go to treat with only information coming from docking studies. So this is the life cycle of the SARS-CoV-2. As we said before, it attaches to the ACE2 receptor with a co-receptor that we know well. It goes into the cytoplasm. It's a single RNA-stranded virus. It goes multiplication. It doesn't go inside the nucleus. That's very important because we save a lot of problems with viruses that don't go inside the nucleus because they cannot enter into a latent phase, and they will be eliminated finally if you can cure the disease. And these are the drugs that can be used in different stages. The most important site of action of all the drugs and antibodies is the entry or the attachment with monoclonal antibodies and many drugs, but we have also protease inhibitors that can block these proteases to get non-structural proteins of the virus. We have remdesivir, which is a nucleotide analog. And all the different drugs, mainly drugs that have no antiviral effect, but anti-inflammatory effect, which is a different target of action. So the first thing that came up is the virus attaches to the angiotensin converting enzyme 2. We have drugs that block this enzyme. Do we have to remove these drugs to prevent the infection, or in subjects who are admitted to the hospital to prevent progression? And the answer is no. Here you have the recommendation of all the international societies. It's quite clear that there's no relationship between being treated with an angiotensin receptor inhibitor and any increased risk of disease. So these drugs have no relationships with disease. And regarding the treatment, we have basically antivirals and immune modulators. Antivirals are drugs that have specifically an antiviral effect, and mainly they are drugs. It is the use of convalescent plasma antibodies and new monoclonal antibodies. That's the main elections we have nowadays. In the future, we'll have peptide-based binders and new drugs, but nowadays we only have this. And immunomodulators, you can block any pathway, as we said before. You can use an interleukin-6 or interleukin-1 receptor blocking agent, a JAK inhibitor, corticosteroids, dexamethasone, as you have seen in the news, and we're going to discuss now. Then anticoagulation is very important. We know this is a procoagulation disease. Deep vein thrombosis, pulmonary embolism, and microthrombosis in all the organs is very common in COVID. So in all the patients, we use prophylaxis with low molecular weight heparin. All the patients, and if they have increased risk, we use full dose heparin. And finally, in the future, we hope to have a vaccine. That's the only thing that can really change the things nowadays. Obviously, when we started to use massively at the same time all over the world repurposed drugs which were used not very commonly, we ran out of these drugs. The prescription was, this is a typical hospital in Barcelona, Hospital Clinic, and this is the prescription rates of tocilizumab, hydroxychloroquine, lopinavir, remdesivir at the beginning of the epidemic in March and April, and all these drugs was out of stock in Barcelona and all over the developed world. That was because everyone started to prescribe these drugs in patients admitted with COVID. Okay, let's start with antivirals. Initially, we thought that the protease of the COVID could be blocked with HIV protease inhibitors. We have two of such inhibitors, lopinavir and darunavir, but immediately it was released a randomized clinical trial coming from China that showed virtually no effect of lopinavir in viral load decay. You can see that with or without, it's virtually the same in mortality or in any other parameter. And Janssen, the developer of darunavir, Janssen Select, did a very nice study, and they were really brave and honest, I must say that. They spontaneously did in the lab study, and they proved that darunavir has no effect in the inhibition of SARS-CoV-2. This should be remdesivir, and this is the comparison of darunavir. They issued a press release informing the physicians that it had no effect. So this is a very honest lab. And immediately the results from the massive UK recovery study with more than 50,000 patients or 16,000 patients, and they informed that there were no differences in death, time to discharge, or proportion of patients discharged alive at 1 month. So there was no effect in the primary or in secondary endpoints. So there's a point where there is a full agreement. HIV protease inhibitors, lopinavir or darunavir, have no role in SARS-CoV-2 treatment. This is something that is quite clear. Let's go with hydroxychloroquine. This is an anti-parasitic drug. It has been a highly controversial issue. It started with a publication from a small French group showing that using hydroxychloroquine plus azithromycin, they could decrease mortality, and they have amazing outcomes. They issued 2 or 3 publications, but in one of them, the so obscure that the society responsible for that publication, the International Society of Antimicrobial Chemistry, issued a statement saying that the publication could not be true, and they asked to remove the publication. Then the New England published a quasi-randomized study. That's something amazing. I had never seen it before. A study can be randomized or non-randomized, but you can be quasi-randomized. So it was something outrageous. And that study initially showed some benefit. And then it came Donald Trump. You know that this is a very controversial man, and he recommended massively the use of hydroxychloroquine to US Americans. He did that. He has been prescribed that drug, and he recommended everyone taking the drug. This had an effect. This is a search to prove the purchases online in US of hydroxychloroquine or chloroquine. And here you have Elon Musk endorsing this drug, Donald Trump endorsing this drug, and this is the number of purchases that it went up amazingly. So this had an effect in the population. Okay, then it came the scientific data. Again, a Chinese study showed it had no effect in the primary endpoint in PCR negativization. An observational study in France, no effect in intensive care unit stays. A second observational study in New York showing no effect in the probability to be intubated and on mechanical ventilation or to die. And finally, an observational study with the Veterans Administration in US, the veterans cohort, that showed again the same things, that it had no effect. So no effect at all. Even a huge study with nearly 100,000 patients hospitalized raised the issue that it could be harmful, it could be toxic. That study was published 22nd of May, and in 1 week, the journal received so many letters saying that the data were false. That forced the editors of the journal to publish an expression of concern, and finally, the paper was retracted in the 4th of June, so in 2 or 3 weeks. That was outrageous. That the journal was Lancet. That didn't happen the same in Lancet and in New England Journal of Medicine, that they were publishing data from a study that was false. It was proved that they didn't have the data that they were publishing. So all the journals started saying that all the research in COVID needed a slowdown in the pace and a better control, a better scientific control that was everywhere. That we were investing a lot of time and resources in drugs that probably didn't have a clear effect in the disease. And there's even a web which is called retractionwatch.com, where you can monitor all the papers that had been retracted in COVID-19 research. And up until now, 36 papers have been retracted definitely and 3 temporarily. So it's something that is really, really controversial. And finally, it came up, the Recovery UK trial, that you know that it's a huge trial that explores many options of treatment. It's a randomized trial that you can be randomized to nothing or to one of all these potential treatments. And this was published in 4th of June when the safety committee recommended unblinding the data and stopping the hydroxychloroquine arm because it had no effect on mortality, which is the primary endpoint, mortality at 28 days, the primary endpoint. So not only this, but it was associated with a higher frequency of invasive mechanical ventilation or death, and it was less likely to be discharged a patient if he was treated with hydroxychloroquine rather than if he was treated with placebo, and that was statistically significant. So hydroxychloroquine at the end of the day was not only not useful, but was even causing harm. So we have analyzed as well in post-exposure prophylaxis, this drug, no effect. In pre-exposure prophylaxis, we did an analysis in our hospital with all the hospital healthcare workers who took the drug. There was no effect of hydroxychloroquine in preventing the disease. And a huge randomized clinical trial has been published now. And shows the same data that we find in the hospital, no effect in pre-exposure prophylaxis. So the final takes with hydroxychloroquine, it has no role at all in the treatment of SARS-CoV-2, even in early treatment, because we have data in pre-exposure prophylaxis or in post-exposure prophylaxis. So everything is concordant regarding the lack of effect of hydroxychloroquine. Let's go with the jewel of the crown, remdesivir. Remdesivir is a prodrug, is an adenosine analog. It has a broad spectrum antiviral activity against many viruses. It didn't have an indication for any one of them so far. And there's a huge difference here. And it is that there is a manufacturer behind the drug that pushes and promotes the data. That changes everything because in hydroxychloroquine, there was no promotion of the drug, so it was more neutral. Here we have a manufacturer. I have to say that in vitro, the initial data showed an effect of remdesivir, so it did show good data in vitro, an antiviral effect and no toxicity, so good data. And we have also data in animals. This is in macaques, non-human macaques, and with the treatment of remdesivir. Oh, please, if you can mute. Oh yeah, thank you. There were lower scores. Oh, sorry. Yeah, we have a lot of noise. If you can mute, thank you very much. There were lower clinical scores in pulmonary pathology, and also an antiviral effect when they look at the viral titers in bronchoalveolar lavage or in lung biopsy. There was a statistically significant difference showing an intrinsic antiviral effect. So this was good initial data. But what happened? The first randomized clinical trial came again from China, and they showed no effect. There were the same need of mechanical ventilation or mortality in those treated randomized to remdesivir or to placebo. That was surprising. And something weird happened here, something awkward. In the same day that this was published, the 29th of April, this was published in New England Journal of Medicine, the manufacturer issued a statement showing a press release with the ACTT results that showed a faster time to recovery with remdesivir. That was unusual because usually you can show a preprint, but usually you don't show a press release of a huge randomized clinical trial. Not only that, but they issued a second press release in the same day, clearly to try to antagonize the negative data of this Chinese study, showing potentially good results from the simple one study that we are going to comment now. Okay, just with this information, with one randomized clinical trial showing no benefit and 2 press releases in 2 days, this was 29th of April, in 2 days, the FDA, the Food and Drug Administration of the US, okayed remdesivir emergency use authorization in COVID. That is something that has been strongly criticized by many scientific associations. Okay, and then we had access to the ACTT remdesivir study. The first publication was an incomplete report. It was called a preliminary report. Actually, it was an incomplete report. It's a huge study. It has 1,000 patients. It's double-blinded, so the highest degree of evidence in medicine. And the safety committee recommended to stop the trial, to unblind the trial, due to a shortened time to recovery in one of the arms. This is very uncommon. A safety committee usually stops a trial when you have an increased mortality or severe toxicity, or when you have a difference in a tough clinical endpoint, so intensive care unit admission, mechanical ventilation, but not in days to recovery only. And actually in the publication, the difference was 11 days to recovery with remdesivir 15 with placebo. That was statistically significant, but there was no benefit in mortality, and that was seen only in patients who receive oxygen, not in patients. You can see it below. This is patients not receiving oxygen, no significant. Patients receiving high flow oxygen or non-invasive mechanical ventilation, no benefit. And patients in the intensive care unit with mechanical ventilation or extracorporeal membrane of oxygen, the ECMO, and there was no benefit either. So only in patients receiving oxygen. This is the number of patients in every group. It's a high number, so this is a strong degree of security that only in patients receiving oxygen in this trial there was a benefit with remdesivir. The good thing is that the drug was safe. Serious adverse events were more or less the same in both arms. And the problem is that the manufacturer believed that this establishes the drug as the reference in this disease, and so futures, as the drug alone has a limited benefit, the interpretation is that future studies will have to evaluate this drug plus a second drug. They said future strategy should evaluate antiviral agents in combination, because with only one antiviral drug, it will not be enough. Actually, this is not what the trial is saying. The only thing the trial is saying is that the benefit of this drug is limited, it's weak. So we need better drugs. And then we have access to the final or the complete report of the study. It took 5 months, which is a lot of time, because the trial was completed. It was just doing the statistical analysis. It took 5 months, and actually there were no major changes. If you see that the more benefit was in patients treated with symptoms less than 10 days, so early, and again in subjects receiving oxygen, but no high flow oxygen, intensive care unit, or mild disease. And in this subset of patients receiving oxygen, they could see a reduction in mortality only in this subset that was statistically significant. Then it comes the simple studies. There are two simple studies, simple 1, simple 2. But the situation, the analysis, the understanding is not so simple. In the simple 1, which is the patients with severe COVID, remember that in the ACTT, in severe COVID, remdesivir had no effect. So in this randomized clinical trial, 400 patients, open label, the study does not have control arm. They are only controlling 5 days versus 10 days of remdesivir. But when they show the data, that is a mortality of 8% with 5 days, 11% with 10 days, or a similar clinical improvement, it's impossible to understand if this is a good thing, if this is a bad thing, because we have no control arms. But the clear thing of the study is that 5 days has the same efficacy as 10 days. That's a good thing, because we could go to just 5 days of remdesivir. And then they did the simple 2 study that was run in patients with moderate COVID. Again, 600 patients, so a huge study, randomized, and this study has a control arm. The study showed no benefit in mortality, 1% with 5 days, 2% with 10 days, or 2% with standard of care, so no benefit. But the clinical status assessed at day 11, which was the primary endpoint, improved significantly with 5 days. This is statistically significant, but not with 10 days. And it's impossible to understand this, because that clearly shows that 5 days is different than 10 days. And actually, it's even worse, because in the arm of 10 days, physicians could give 5 to 10 days, whatever they believed. And the median days of treatment was 6. So we are comparing an arm which was treated with 5 days to another that was treated with 6 days. One has a significant benefit in the primary endpoint and the other one has not. So in summary, the simple 1 shows that 5 or 10 days is the same, and the simple 2 shows that 5 days is significantly different than 10 days. Even if you see the daily discharge rates of the patients, in red you can see standard of care, This is the number of days hospitalized. And you can see that those who were treated with 5 days, there's a peak of discharge at day 6 or 5 or 6. And those who were treated with 10 days, there's a peak of discharge at day 10 or 11, suggesting that discharges were delayed to allow the completion of treatment. So it could be that treatment delayed the discharge in the hospital. What's the conclusion of the authors? This is the first time that the authors are honest, and in the conclusion in the paper in JAMA, they actually show that 10 days had no benefit, 5 days had only benefit in the clinical status, but not in mortality or any tough endpoint, and the difference was of uncertain clinical importance. And I think this is the right thing to say. And very recently, I think it has been 2 or 3 weeks ago, the WHO sponsored clinical trial, which is the Solidarity trial. This is a trial that has the primary endpoint in mortality. I think this is the right thing to do with COVID. We cannot copy the things we did with influenza, going only to a score of clinical improvement as the main primary endpoint. We have to go to clinical tough endpoints: mortality, intensive care unit admission, mechanical ventilation. So the WHO recommended doing trials with mortality. This is a huge trial with 11,000 adults, and more than 2,700 were randomized to remdesivir, and we have a control arm of 4,000 patients. The primary endpoint is 28-day mortality, and it was 12% over the trial, 39% in those who were ventilated, and 10% in those who were not ventilated. So mortality is high, and with this number of patients, it allows a robust comparison. And in this comparison, you can see that virtually there is no difference at all with remdesivir and the control. So there was no difference, and that's definitive in mortality in this study with remdesivir. That went into the press immediately because this is a very sensitive issue that remdesivir showed no effect in mortality. And the WHO did a meta-analysis, and you can see that the final data, it's not statistically significant. It crosses the 1, so you cannot say that the drug improves mortality, but in the subset of subjects receiving low oxygen flow, it could have a mortality benefit. It could. You see that it's not, this is the final analysis compilation. It's not statistically significant, but it could be a benefit only in this subgroup of patients. So the final takes, I would say that remdesivir is the only antiviral with some but inconsistent efficacy report so far. It has highly contradictory data across studies with low level of final evidence, and the benefit is of uncertain clinical importance. It could shorten the time to recovery only in moderate COVID, and it has no benefit in mild or severe disease. Okay, it has been included in all treatment protocols all over the world, in all Europe. It's included in all treatment protocols of patients admitted with moderate COVID. That's the only indication. It has been approved with a final approval by the Food and Drug Administration last week. The emergency use authorization was issued in May, but the full approval was last week. This has been very controversial because this approval might clog the pipeline of other drugs and may have unwanted effects on research with any other drugs, because the emergency use authorization for a disease can only be given once. Once you have a drug approved, you cannot grant another emergency use authorization to a second or a third drug. So no more drugs are going to get this approval. It would be or should be the default arm in all research clinical trials, even when the efficacy of the drug is very debatable. Any trial you do, any company does, will need probably a remdesivir control arm and will need the collaboration of the manufacturer of remdesivir. And actually, in the plan of development of the drug, in the ACT2, they have 2 arms with remdesivir baricitinib against remdesivir, but they do not have a control arm. So that could be a big problem in the future. And the developer of remdesivir, as you know, has started a phase 1 clinical trial to evaluate the efficacy of inhaled remdesivir. This is the final take in Twitter. which is one of the ways to get the scientific information these days, sometimes more rigorous than medical journals. So it's very important. This is Boguma Titanji, a scientist from Emory University in the US, and this is her final take of remdesivir, when it's beneficial or not. Okay, just to let you know that there are other drugs, other repurposed drugs that are being analyzed, including 2 antiparasitic drugs, ivermectin and nitazoxamide. This one, the second one, having a randomized clinical trial, double-blind trial, 400 patients in Brazil showing faster clinical improvement and even an antiviral intrinsic effect with faster biological resolution. So we'll see. It's just one clinical trial, but we'll see. We have trials with anakinra, which is an interleukin-1 antagonist, acalabrutinib, which is a Bruton tyrosine kinase inhibitor, and ruxolitinib, which is a JAK inhibitor. These are small trials. Sometimes they have no control arm, but all this is being analyzed, and we'll see in the future if they have any benefit or not. What about monoclonal antibodies? We have some monoclonal antibodies that could have interest in COVID. This is a nice summary. As antibodies don't go inside the cell, we usually use antibodies to inhibit the attachment or the entry of the COVID. But they could also have other mechanisms of action. Mainly they are antivirals, but some of them are also immunomodulators. So a huge effort is being done with monoclonal antibodies in all the medicine, but also in COVID-19. What happens in this disease is that humans get cured of COVID. So if they don't die and they cure, they develop antibodies. When we cure of COVID, we develop more than 40 antibodies, any individual. Of them, 3 or 4 are neutralizing and can be copied. So what we want to do is copy those antibodies and give directly the antibody. We can identify the antibodies. We know which antibody has a neutralizing activity, and we are going to try to copy that antibody. Obviously, the virus can mutate. This is a wild type virus. This is the nice inhibition with these 3 antibodies in 3 different colors. But with this mutation or this mutation, we can see that 2 antibodies here or 1 antibody here in green lose the activity. So the virus has the possibility of selecting a mutation and developing resistance against a single antibody. So the best thing to do is going to be administering 2 or more antibodies, what we call a cocktail of antibodies. And remember this word nanobodies, which are smaller antibodies that has been discovered in camelids, in camels and llamas, and also in sharks as well. They are basically smaller antibodies because they have no light change, but they perform the same. And they are easier to construct in the lab, and they are being developed to treat COVID, the nanobodies. You'll hear this word a lot in the near future. There are 2 companies involved with antibodies. The first one is Lilly. This company has developed an IgG1. They have done a randomized double-blind phase 2 study. This is the viral load decay. Everything looks very nice. This is a 4-log decrease in 11 days, amazing. But be careful because the red one is the placebo. So subjects spontaneously show a decrease of 4 logs in 10 days. So what we're going to analyze is the difference from placebo to the antibodies. And you can see that the difference is not so big. It's statistically significant, but it's not so big. It's half a log. And that's a small difference. Okay, they use 2 antibodies, bamlanivimab and etesevimab, 2 monoclonal antibodies. They use the combination, and they have issued a press release, and just a press release saying that they met the primary and secondary endpoints, that the improvement in the clinical picture and in hospitalizations or emergency room visits was statistically significant, and that was amazing. There's a full program of development, but don't be so happy because just yesterday or 2 days ago, they issued a press release saying that in the only trial, which is ACTIV-3, done by the National Institutes of Health in the US evaluating bamlanivimab in hospitalized patients with severe disease, the antibodies failed and the trial has been stopped for futility. So they will only continue to develop these antibodies in mild to moderate or ambulatory recently diagnosed patients or in prophylaxis, but not in patients hospitalized with moderate or severe disease. And the second company is Regeneron. Regeneron has as well a couple of antibodies. It's virtually the same. Using two antibodies, you impede the development of mutations. And they have issued a press release again to describe the first 275 subjects enrolled. And they have showed something which is very nice because it's science. And it's that the serological status, so the level of immunoglobulins of the subjects, correlated with the viral load. So the more antibodies you have, the lower is your viral load in COVID disease. And that's nice to know. And there's as well a correlation with clinical pictures. So with clinical alleviation, once you develop the antibodies, you get cured and your viral load goes down. That's okay. But in addition of this, they met the primary endpoint of a greater viral load reduction with some of the doses, the high dose, and that's important as well. Okay, we'll see how this goes because the program includes more than 2,000 people. It's just enrolling now. We'll see the data with these antibodies. And finally, we go with immunomodulators. Immunomodulators have no antiviral activity. They are only directed of improving this exaggerated immune response. The most used is tocilizumab. Obviously, these go to critically ill patients with COVID. This is one randomized clinical trial. This is a cohort in the US, an important cohort. Obviously, there were differences in the baseline because those who were treated with tocilizumab had a worse condition, but they tried to adjust this in the statistical analysis, and they reported a lower risk of death. The difference in the mortality was significantly different, and that is good, although obviously residual confounding could exist in the baseline. And another cohort, a second cohort, confirmed a reduction, an important reduction, 45%, that's important, in the risk of death in critically ill patients with COVID. What happened? Three in-line research, randomized clinical trial, open level, and double-blind with 250 patients have reported the primary endpoints of clinical progression, entry into the intensive care unit with mechanical ventilation death or aggravation of the clinical picture or intubation or death, and the three randomized clinical trials have failed in a row. 3 randomized clinical trials. And the owner of tocilizumab, that is Roche, has a known phase 3 program development with 2 clinical trials in subjects with COVID-associated pneumonia hospitalized. It's the highest level of evidence. It's a double-blind randomized clinical trial controlled with placebo. In the first one, fully powered trial, they did not meet its primary endpoint of improving the clinical status or reduce mortality or differences in ventilator-free days. So overall, there was no benefit for the patients. But in the second one, in patients that were not so severe, using the same drug and the same dose, they proved an improvement in the primary endpoint, which was very similar, the improvement in the risk of going to mechanical ventilation or death. So a bit of discordant results, despite there was, even in this second trial, there was no improvement in the time to hospital discharge or improvement in the clinical status or overall mortality, only in this analysis of progression to mechanical ventilation or death. This is quite disturbing, and you have to understand why is that difference in studies raised by the same company similarly in patients with theoretical similar inclusion criteria. And maybe the difference is that the risk of mechanical ventilation at baseline was 37% in the Covacta, the one who failed, and 0% the risk to go into mechanical ventilation in the EMPACTA. So maybe that the conclusion is that tocilizumab has no effect if you go too late to treat the disease. So in the final takes, I would say that the data are inconsistent. Many studies have shown no benefit, including some of the randomized clinical trials, including double-blind ones, which is the highest level of evidence in medicine. But two twin studies, as you have seen, showed discordant or even contrary results. So we're going to see if we can understand this. Maybe what we could say is that tocilizumab must not be given too late, because if you go too late to the intensive care unit, you're not going to get any benefit at all. I could say that more data are needed, but honestly, with the data we have, we should be able to understand the discordant effect of the drug, which is difficult to understand so far right now. We got corticosteroids, and this is probably one of the most important trials in COVID. The recovery trial reporting that dexamethasone reduced mortality. This is the first randomized clinical trial that shows a benefit in mortality, which is robust. This is the number of patients randomized, which is huge, and there's an improvement, and it's concordant in patients with mechanical ventilation, receiving oxygen or receiving respiratory support. You can see there's a benefit. So the only situation where there's no benefit in subjects that don't need oxygen. So if you go very early to mild disease, there's no role for corticosteroids in that scenario, but in moderate or severe patients, there's a benefit in mortality. That's very important. This is not clinical improvement or time to discharge, but this is mortality. There's a second trial in Brazil confirming this. This trial was done in intensive care units, and then the benefit was much more limited. Was statistical in some endpoints, but the benefit in mortality was much limited, because probably it's too late if you go to the intensive care unit. The same happened in the Cape COVID in France, another randomized clinical trial done in intensive care units. So if you have to use any corticoid, see that this is hydrocortisone. It's not dexamethasone, but it's similar. So don't wait that the patient to be admitted to the intensive care unit. And even in this part of the treatment, which is the part with more robust data, there are discordant studies. We have, again, a randomized clinical trial showing no benefit in mortality. These patients were not in the intensive care unit, and there was no benefit in mechanical ventilation, in duration of hospitalization, or that's expected in virus clearance, because corticoids that have no effect in virus clearance. So even with corticoids, there's a small controversy. So we have here a WHO meta-analysis, and you can see that the final effect is a benefit in mortality. Again, it's in the limit of statistical signification with all the information that has been raised so far. And subjects that are receiving vasoactive medications, so that have shock, they do not show any improvement because probably it's too late. But patients that do not receive vasoactive medication, they show a significant benefit. So with corticoids, I would say that the final take is that this is the drug with the highest level of evidence in COVID and consistent. So it's not complete, as you have seen in all the studies, but they can reduce mortality in both moderate and severe pneumonia, not in life-threatening pneumonia. That's important, and not in mild COVID. So we are not going to treat with corticoid subject with mild COVID. These drugs are approved. They are cheap, safe. They can be given orally. All the ongoing studies have been interrupted because the power of the solidarity is huge. It's impossible to go against this trial. And corticoids or dexamethasone, as an example, are included in all the treatment protocols. And importantly, don't go too late with corticoids. And the last thing is convalescent plasma. Convalescent plasma has been used a lot in many diseases, infectious diseases with no treatment. And it's very easy. If humans cure of a disease and they develop antibodies, you can get those antibodies and transfuse them to very ill patients and see if there's an improvement. And I must say that the animal models were good. So if you use high dose antibodies low-dose antibodies in the convalescent plasma or nothing at all in control, there's a significant difference in the histopathological lesions and as well in the viral load. This is the viral load in bronchoalveolar lavage at 5 days. No virus at all in those macaques or mouse that received high or low dose of convalescent plasma. So it has good data in the animal models. US love convalescent plasma. This is a preliminary report that includes 20,000 patients that have received convalescent plasma in US, and they report safety just in this publication of Annals of Internal Medicine, and they report that it is safe. Okay, it is safe. What about the efficacy? In the first analysis of the expanded access program with 35,000 patients treated, mainly intensive care unit or receiving mechanical ventilation, the transfusion of convalescent plasma with high antibody levels in hospitalized patients reduced mortality. And transfusions that were given in the first 3 days of clinic had greater reductions in mortality than when these transfusions were done later on. And the difference was statistically significant. That's a good data. In a second study shows virtually the same. That is the efficacy of all the patients receiving convalescent plasma. That's the efficacy of those transfused in the first 72 hours or in the or beyond 72 hours. And this is the efficacy in patients that receive a high dose of neutralizing antibodies in the first 72 hours. And that's the biggest benefit. So it's important that this is science, and probably if the convalescent plasma has any efficacy, it will be only in the first days and in subjects receiving a high dose of plasma, including antibodies. A study coming from China, a randomized trial, which is not clear in the results, showed a benefit in subjects with severe disease, that's statistically significant, but not in subjects with life-threatening disease, showing the same as with tocilizumab. If you give these treatments too late, they are not going to give any benefit in mortality. And a randomized clinical trial in India has failed. And it's difficult to understand why this trial failed. There's no benefit in a composite endpoint of progression to severe disease, mortality, mortality analyzed separately, or mechanical ventilation, or in biomarkers of cytokines, LDH, D-dimer, C-reactive protein, ferritin. There was no benefit in biomarkers. So the best thing we can do today with convalescent plasma is wait for the clinical trials that are ongoing. And in this meta-analysis, the final take is that in the fixed effect model, there's a significant benefit in mortality favoring convalescent plasma. So the final take of convalescent plasma should be that this is a scientifically sound strategy that has been tried in many other infectious diseases with no treatment. The initial data suggests that it could have a role in severe COVID when it's administered early and when the plasma contains a high level of neutralizing antibodies. And anyway, it looks like a safe strategy. Okay. So that's all I wanted to tell you today. What are we doing today in the hospital? And I would say in all the hospitals in Spain or in Europe, this is in the treatment of subjects with COVID. I'm working nowadays in the COVID ward in my hospital. These are tough times for us physicians. We give dexamethasone, as we said before, in subjects with moderate and severe disease, including those in the intensive care unit. Remdesivir only to subjects with moderate disease. That means lung infiltrates plus low saturation of oxygen, but not in the intensive care unit or mechanical ventilation, because in these situations, we know it has no benefit at all. Tocilizumab, if you have moderate pneumonia, don't wait too long. We are not sure if this is having an effect or not, but in subjects with moderate pneumonia with markers of a strong inflammation, we give tocilizumab. All subjects receive prophylaxis with heparin, and we don't give antibiotics only if there's a suspicion of a bacterial infections. And drugs with no effect have been removed from all the protocols. So the final takes of all this analysis, which is not easy at all, I would say that with the COVID epidemic, we have learned a lot of new things. The first one is that the top medical journals, including New England and Lancet, and the US health agencies, including the Food and Drug Administration and the Centers of Disease Control, have lost the rigor that they usually had in the past, and they have been a collateral victim of COVID-19, and this has been very sad to be seen by us researchers. Regarding the antivirals, the efficacy of drug reproposing has been very limited. We have a great hope with new design drugs and with some monoclonal antibiotics that are going to come in the near future. These monoclonal antibodies and nanobodies are a very exciting pipeline. We're going to see a lot of the development in the research regarding this. They have a high affinity for the spike. They are amenable for long-acting activity, and they are going to be used as cocktails because a single antibody, it's relatively easy for the virus to develop a mutation and get resistance. Regarding the immunomodulators, I would say that dexamethasone works. It's a strong word. It works in moderate to severe disease. Tocilizumab is uncertain. I would say that it's still uncertain. And we have promising data with many drugs that are going to see in the near future. And regarding the unknowns, we don't know if these treatments will be useful for the next coronavirus pandemic because the coronavirus is mutating and evolving slowly. We'll see. And we don't know as well if the widespread use of these antivirals has the potential to develop resistance in the epidemic of SARS-CoV-2. That's all, and let's wait for vaccines because the COVID-19 vaccine, I would say it's the best scientific race of the decade. The competition is huge. You're going to see news every day in the media, in the TV, in the newspapers. All the companies involved leak news. Sometimes I wouldn't say fake news, but news that do not merit to be promoted through the mass media like phase 1 data or phase 2 data. We are going to wait the phase 3 data of the COVID vaccine. Okay, thank you very much. That's all. Sorry for being a bit late, and I'm happy to receive any questions if you want regarding the treatment or any other field of the COVID disease.
Hi, hello.
Yeah,
Can you hear me?
Yeah, that's
okay. Because I had problems with my microphone. Thanks so much, Jose Maria, for your amazing presentation. Has been really great. It has been a pleasure to listen to you. So you can hear me? I'm not sure.
Yeah,
okay.
I'm gonna go to the easy ones. Manikanda Sur asks if is there any treatment for the loss of smell and taste of COVID, and the answer is no. It usually recovers spontaneously, but it takes weeks, as you say. 2 weeks after the infection, he regained a bit of that. Yeah, probably you will recover everything, but it takes some weeks or months. Ruth Villa asks about medical teams and researchers worldwide. Has it really worked? Oh, and she says that was a fantastic presentation. Thank you, Ruth. Well, you must understand that this is a competition. It's a lot of money with patents and sales. So I would say that scientists are sharing most of the data, but obviously those who have a new amazing antibody or a new amazing of a mechanism of vaccine are probably not going to share all the knowledge. And I think that's understandable. So there's a collaboration between companies, but I wouldn't say they are sharing all the information. R.A. Pavintra asks if there's any chance of reinfection. Yes, it is. Yes, it is. That's a good question. You can get reinfection by two ways. The first one is losing your immunity, your cells or your antibodies. And the second one is if the virus is able to change so much that your immune response does not no longer recognize the virus. That can happen. It has been reported. So far, it's extremely rare, but we'll see in the future because it could be. It has only spanned 5 months from the first wave to the second wave, so we'll see with more months if that is more common or not. But anyway, it has been reported. Patients with two different viruses in the first and in the second episode being reinfected despite having a theoretically protective level of antibodies. Let me see more questions. Oh, Maria Moscoso asks about a long COVID. Yeah, that's a good question. Long COVID is the clinical symptoms that persist after being recovered from COVID. It happens. It has been documented. It's not uncommon. So once you have COVID and you recover, some patients feel with a loss of strength. Some have low-grade fever, have symptoms, and they have autoantibodies. So when we look at autoantibodies, anti-nuclear antibodies, they have positive values. So it's quite clear that in some cases, the antibodies we develop against COVID, remember that I said that once you recover from COVID, you develop about 40 or 50 antibodies. Sometimes some of these antibodies have anti-nuclear activity. So they are they have an autoantibody effect, and that can be dangerous. We don't know yet what to do with these subjects. They do not have an involvement of any organ, so they have no involvement of the kidney, of the joints, of the lung, but they feel tired, and it's not something psychological or psychiatric, but it's something related with autoimmunity. We'll see. We'll see. There's one question regarding vitamin D coming from Laura Rebel. There's an association between low levels of vitamin D and the risk of infection by any severe disease. What happens is that when you have a depletion of vitamin D, you usually have an illness associated with that. And probably that's a bias in the analysis. So it's like saying if you are ill in the bed because you have a severe disease, you have a higher risk of getting COVID. And if you get COVID, progressing to a severe disease. It's not true that's related with the deficit of vitamin D, and that if we give vitamin D to the patients, they are going to recover. That's not clear at all. My opinion is that a bias between the chronic condition that caused the vitamin D deficit. There's a question from Maria Moscoso about the timings of the vaccine trials. He or she says, there seems to be a big rush from governments regarding this issue. Yes, you are absolutely right. All the news that you're going to see in the press are fake regarding vaccines. The first, you can go to the web of the WHO. You have all the phase 3 of the vaccines trials over there, and you have the date when they are supposed to finish the trial. They are starting to close at December of this year, but most of them are going to close in March or April 2021. That means that in that date, they would know, they would close the trial, they would need 1 or 2 months to do the analysis and know if the vaccine is protective or not. So we're not going to have news before the spring of the next year, 2021. At that time, we're going to know the primary effect of the trial. So if the vaccine is safe and if it's protective or not. We have never found a vaccine at the first attempt in the past. So it should be quite unusual that the first trial with the first vaccine is successful. That should be quite unusual. It could happen, but I wouldn't expect that the first trial shows a protective vaccine effect and it's going to be commercialized immediately. As you all know, all the companies that are running phase 3 trials are producing the vaccine in the factories. So they are doing the production at risk. They don't know if it will work or not, but they are producing millions of doses of the vaccines. Many companies, AstraZeneca, GSK, Pfizer, so Janssen. The problem is if you finish your study, let's say in February 2021, and you are very lucky and the vaccine works, and then you have to start the production, it's going to take 3 or 6 months to get millions of doses of the vaccine. And then you have to send the vaccine to everywhere, and then you have to administer the vaccine to all the patients. So that should take many doing production risk. That means that they have millions of doses in the lab, and if the vaccine trial fails, they are going to throw away all that vaccines because they are not going to work. That's a risky strategy, and that's a risky decision. We'll see. Jaya Dipti Gutula says, thank you very much for this clear presentation. Thank you very much, Jaya. Oh yeah, Susana Rique asks about the different mortality rates in different countries. What's the reason? Yeah, there are many reasons, but mainly the reasons is in the way they analyze the numbers. Obviously, when you look at the mortality rates of Spain and Germany, they are 10 times higher in Spain. That the difference is huge. Probably most of the difference because the hospitals are the same, the treatments we give are the same. The number of patients or citizens with infection is somewhat different, but the mortality rates should be more or less the same. So it's mainly the way when you analyze the data. For instance, in Spain, if you die with a positive PCR, your cause of death is COVID, even if you die from a neoplasia or a transplant rejection. In other countries, if you die with a positive PCR, but your cause of death is a cardiac arrest, you're not counted as a COVID death. So that's the reason for the difference. More questions? I'm not sure if I am missing any other question.
Maybe it was at the beginning.
Oh, yeah. I'm very sorry to give you so much information in so reduced time. I'm very sorry about that.
It has been great, and I think we have taken the most, and we will have also the recording so we can digest all the amount of information. So it has been great. The first one that it's in red on the top, it's from Aramis. Still minute.
I don't see it.
She's asking about pyroptosis. Does it mean that in COVID-19, what it's happened, it's an overreaction of first barrier of immune system?
Yeah, it is. Well, in the beginning, in the first wave, we didn't know nothing at all about the COVID. Patients were dying in the hospital, and they have increased levels of interleukin-6, ferritin, PCR. So they had an inflammatory response many, many days after the infection. So it seemed that the infection had been cured, and they had an inflammatory disease. And so we thought it should be a good idea to give something to reduce the inflammatory response. We started with corticoids, and then when we knew that the interleukin-6 was in the middle of all this cytokine release syndrome, as we had interleukin-6 antagonists or blockers, we started giving that to the patients. It was impossible to understand if that was being useful or not, because we were giving to the patients lopinavir, hydroxychloroquine, interferon, tocilizumab, corticoids, antibiotics, everything, because they were dying, sometimes young patients. So until we had randomized clinical trials, which is the only way to understand things in medicine, we could not understand what was useful and what not. And this pyroptosis, or this exaggerated or dangerous immune response to a pathogen, to a virus, probably is not so important as in the past. The proof is that when you see the results of convalescent plasma or tocilizumab, they are not so clear. So I agree that if you give them too late when you are in the intensive care unit, it's difficult to see a benefit in mortality, but the benefit is not so clear. So probably that's an epiphenomenon, not so important as we thought.
Okay, there is another question regarding about the efficacy of Tamiflu. Has been tested in COVID?
Yeah, there was one trial and it was not efficacious. Yeah.
Okay. So I think that you covered almost all questions. I don't know if there has been anything. There is a lot of congratulations for you.
That's good.
Here we have one, maybe it could be the last one because we are running out of time. We appreciate dedication. What's your opinion about new dominant viral mutations emerging in different countries?
Yeah, that's amazing. In the beginning, we were said that the beta coronaviruses were not able to mutate a lot. We were said that HIV or the influenza virus mutate a lot, but the coronaviruses, the ability to mutate was very slow. And they said one example is that the first epidemic in 2003 took 10 years to be able to provoke a second epidemic with the MERS in 2010 in the Middle East. And then it took 10 more years to be able to produce the SARS-CoV-2 in 2019-20. So that was like a proof that the ability to mutate of the coronavirus was very, very limited. That was not true, because the first mutation in a position was seen only 2 months after the first epidemic. And not only that mutation was isolated in some samples, but it became dominant strain of coronavirus all over the world. So that mutation allowed the virus, and it has been proven in the lab, to be more efficient in the transmission. It was more infectious and more efficient in the transmission. Luckily for us, it was not more severe. That's a good news. So that mutation increased the fitness of the virus, but was no more dangerous. But as it was more fit than the wild type, it replaced the wild type in 100% of the samples all over the year. Today we saw in the news new mutations in Spain and Europe, and we're going to see this in the near future. So far, they have no impact in the disease in the clinic, but they could have an impact, for instance, in the vaccine, in reinfections, as you asked before, and in the efficacy of some drugs, for instance, monoclonal antibodies, because monoclonal antibodies are designed to a part of the spike. If the spike mutates, it could be that the antibody is no longer attached to the receptor. So we're going to see in the future if this impacts or no. So far, it's only something we see in the epidemiology.
I don't know if you still have some minutes. There is a particular question from Jaya in India. She said, if a patient with COVID-19 and severe pneumonia, who is healthy otherwise, was at stage 2 and has been treated with remdesivir and got recovered, but his HbA1c levels were abnormal, which led to diabetics. After 2 months of treatment, their tomography chest scan was again the same, with represented CORADS 5. I'm sorry, I don't know what it means, but the patient had no symptoms. How long for the lungs back treatment symptoms or adverse events which we should be worried about?
Yeah, that's a good question. There can be a sequela in the lung after recovering from a coronavirus. You can have a pulmonary fibrosis. So it's not only an infection that gets cured and the lung recovers to normal state, but sometimes you can have a scar of the lung. You can have a scar and a pulmonary fibrosis. That's a line of research today, The way to research this is with a CT, pulmonary scan, with a functional test to breathe the patient, or even a pulmonary biopsy. And some patients have their lungs with fibrosis, with the limitations of the ability to do sports or even normal life. So some of them need oxygen, and that takes months to recover, and very, very special cases, only in those who have been admitted in the intensive care unit for long, they can have pulmonary sequelae all their life.
Okay, thank you. Perhaps we should go to the last one because this is never finishing. Is there any kind of panel of biomarkers apart from the D-dimer being tested at the moment of patient's admission to guide on the treatment implementation and prognosis?
Oh, I see it. It's from Coya Dulsat. Yeah, we have a list of biomarkers, and we have even formulas. We have scores. We have developed a list of scores that the idea is to stratify patients when they are seen in the emergency room. So in other ways, the first time you see the patient, try to stratify the patients who are going to have a bad evolution, send them to the hospital, and be hesitant if they are going to need an admission to the intensive care unit. The list is very well known: ferritin, fibrinogen, interleukin-6, C-reactive protein. So all these, the D-dimer, maybe it's the most important, and some scores have been published, and just giving a score to every one of them gives you the risk of the patient. Obviously, the higher the levels of all these biomarkers, the higher your inflammatory response, activity of the disease, and your risk of progression. Yeah.
Okay. Thank you very much again, Josep. It has been great, all this session. I think we can stop here. You see all the congratulations for everybody because this amazing talk, I think we have learned a lot. And
That's
thanks
good.
so much. Thanks
Many
everybody for joining. Okay. Bye-bye.
Okay, bye-bye. Thank you very much. Bye-bye.
Thanks very much.
Thank
Bye.
you.
Thank you.
Bye.
Thank you.
Gràcies, moltes gràcies.